Hey all, welcome to Sharetreck. This is Raj here. Today's topic is design principles in synthetic biology. Hello everyone, and welcome back to our series on synthetic biology. This is the third episode of the mini-series on synthetic biology basics for investors. Synthetic biology market is growing quickly, and with it, the prospects of great investment opportunities, of course, are coming up. And at the start of any industry, there is a lack of information, and therefore there is a higher risk. And the investor needs to put some efforts to understand a little bit about the field before proceeding. And that's the reason why at Sharetreck, I'm working on putting together basic information that an investor might need in order to reduce risk of investing in the emerging field of synthetic biology companies. I have many more upcoming episodes in this series, and I provide a good understanding of synthetic biology from an investor perspective. We're not going too deep into it like science students, but just deep enough to understand what the companies are saying, the ones that we are planning to invest in. So if you're interested, please press the subscribe and notify button so that you do not miss any of these episodes going forward. With that said, let's get started. Welcome back, friends. In the previous episodes, we discussed the fundamental role of DNA in synthetic biology. Today, we'll be discussing the three main design principles that are used to create new biological systems. One of the key design principles in synthetic biology is modularity. Modularity refers to the use of interchangeable parts that can be combined in different ways to create new biological systems. One of the key concepts in synthetic biology is modularity, which refers to the idea that biological systems can be designed and constructed from interchangeable parts or modules. We use a similar concept in software engineering where we create modules that are reusable. Modularity is important because it allows synthetic biologists to create complex systems by combining simple standardized building blocks in different ways, and then assembling them as required in order to perform a particular function. This approach is similar to the way the electronic circuits are designed, with individual components such as resistors, capacitors, and transistors doing different functions, but they are combined together in different configurations to create a wide range of circuits with different functions. In synthetic biology, modular design is typically achieved through the use of standardized genetic parts such as promoters, coding sequences, and terminators. These parts are designed to be interchangeable and easily combined, allowing synthetic biologists to create genetic circuits with specific functions. For example, imagine that you want to create a genetic circuit that responds to a specific signal, such as a particular chemical in the environment. You might start by selecting a promoter that is known to be responsive to that chemical, and then combine it with a coding sequence that produces a protein in response to the promoter. You could then add additional modules to the circuit to amplify the response, regulate the timing of the response, or connect it to other circuits in a larger network. By using modular design principles, synthetic biologists can create complex biological systems with precise control over their behavior. This approach has led to the development of a wide range of synthetic biological systems, including biosensors, drug delivery systems, and even organisms with novel functions. Overall, modularity is a powerful tool for synthetic biology design, allowing the researchers to create complex biological systems with greater degree of precision, efficiency, and flexibility. Another important concept in synthetic biology design is abstraction, which refers to the process of simplifying complex biological systems into simpler, more abstract models that can be more easily understood and manipulated. In other words, the complexity is encapsulated into a layer of simplicity so that the user just needs to know the function. The user does not need to know how the function is achieved. So that's another way I would describe abstraction. Abstraction is important in synthetic biology because biological systems are incredibly complex with many different components and interactions that are difficult to understand and predict. By abstracting these systems into smaller modules and simpler modules, synthetic biologists can more easily analyze and design them and create new systems with specific functions. There are several levels of abstractions in synthetic biology, each of which simplifies the system in a different way. At the highest level, synthetic biologists might model a biological system as a set of input and output functions without worrying about the underlying biological mechanisms. This is sometimes referred to as black box modeling. Because the details of the systems are hidden inside the black box, you just don't need to know what is in the black box. You need to know what goes in and what comes out. At the next level of abstraction, synthetic biologists might model a system as a set of interconnected genetic circuits, each with specific inputs, outputs, and functions. These circuits might be designed using modular principles as described in my previous answer. So, as I was discussing with you before, in the field of software, this system has been used. Earlier, we used to have a discipline called as Structured Systems Analysis and Design, SSAD, in which we always used to talk about what is called as a context analysis diagram, where we used to have a circle to show something happens inside it, and then we show the input and the output, and then we start digging deeper into it by breaking down into modules and connecting those modules in a sequence, and then further breaking down the modules into individual functions. So this is similar to that. At the lowest level of abstraction, synthetic biologists might model a system using detailed mathematical models that stimulate the behavior of individual molecules and reactions. This approach, which is sometimes referred to as bottom-up modeling, allows synthetic biologists to understand the detailed mechanisms underlying the biological systems and to predict their behavior under different conditions. Overall, abstraction is a powerful tool for synthetic biology design, allowing researchers to simplify complex biological systems into more manageable models and to create new systems with specific functions. This approach has led to the development of a wide range of synthetic biological systems, from simple genetic circuits to complex organisms with novel functions. By breaking down complex systems into their component parts, researchers can better understand how these parts work together to create the whole. A third design principle is standardization. Standardization involves using common set of rules and protocols to ensure that biological systems are constructed consistently and can be easily shared between researchers. For example, in the discipline of software, we have a protocol of how to name data elements, how to name modules, so that everybody understands what to look for and where. Standardization is important in synthetic biology because biological systems are incredibly complex, with many different components and interactions that can vary widely between organisms and experimental setups. By using standardized genetic parts and methods, synthetic biologists can reduce the variability and increase the reproducibility of their experiments. So unless you can consistently reproduce the output of the experiments, you cannot be confident that what you have is a foolproof system. And they can then more easily share the results with others, knowing that the others can also replicate it reliably and get the same results. One of the most important examples of standardization in synthetic biology is the use of standardized genetic parts such as promoters, coding sequences, and terminators. These parts are designed to be interchangeable and easily combined, allowing synthetic biologists to create genetic circuits with specific functions. Standardized genetic parts are typically designed using common DNA sequence and cloning methods, which enable easy sharing and replication of the parts among different labs and researchers. In addition to standardized genetic parts, synthetic biologists also use standardized methods and protocols for genetic engineering, such as PCR, gel electrophoresis, and DNA sequencing. These methods are well established and widely used in molecular biology, and their standardization enables easier comparison of experimental results and more efficient transfer of knowledge and technology between labs. Overall, standardization is a powerful tool for synthetic biology design, allowing researchers to reduce the variability and increase the reproducibility of their experiments, and then more easily share their results and methods with others. This approach has led to development of a more efficient and collaborative synthetic biology community and has enabled the rapid progress and growth of the field. By using these design principles, synthetic biologists are able to create complex biological systems with specific functions. In the next episode, we'll be discussing the ethical and societal implications of synthetic biology. If you are a genomics investor, you would find these videos very helpful, and you can revisit them anytime to brush up on concepts. So please make sure you subscribe and press the notify button so that you do not miss the next episodes. You can always go to our homepage on YouTube and search for the episodes. I've also made a separate playlist for synthetic biology. So if you go into that playlist, you will find all our episodes in sequence, and you can watch them again if you want. Thank you for joining me today, and I look forward to seeing you again the next time. Bye for now.